Hello young men, you are welcome in the zoology class organized by Jan Vikas Mahasabha and Society of Career Technology. Today, before coming on the topic, I would like to share with you some questions also on the topics which we have studied. For example, in some of the examinations you get objective questions or multiple choice questions. They are very important. Let us have a glimpse of that. For example, who gave the cell theory? Schleiden and Schwann, Watson and Crick, Darwin and Wallace, Mendel and Morgan. Second, which part is not found in a prokaryotic cell? Nuclear membrane, cytoplasm, cell wall, plasma membrane. Third, a unit membrane is made up of phosphoprotein, protein and lipids, phospholipids, cellulose. Question 4, who gave the word cell? Leeuwenhoek, Robert Hooke, Fleming, Robert Brown. Fifth, in which part of the cell food liberates energy? Chloroplast, mitochondria, nucleus, nucleolus. Question 6, who discovered mitochondria for the first time? Colliker, Robert Brown, Altman, Robert Hooke. Uracil is found in DNA, RNA, DNA and RNA or protein. You can note the answers. The cell theory was given by Schleiden and Schwann. Question number 1A. 2. Which part is not found in a prokaryotic cell? The nuclear membrane. 2A. Third, a unit membrane is made up of B. Protein and lipids. Fourth, who gave the word cell? Answer is B. Robert Hooke. In which part of the cell food liberates energy? Mitochondria. Question 5B. Who discovered mitochondria for the first time? Colliker. Question 6A. Uracil is found in RNA. 7B. Similarly, some more questions. What is the powerhouse of the cell? Chloroplast, mitochondria, Golgi body, nucleus. 9. What is suicide bag of cell? Lysosome, ribosome, mitochondria, nucleus. 10. What is synthesized in ribosome? Protein, fat, carbohydrate, lipids. What is physical basis of life? Nucleus, cell, protoplasm, food. 12. True nucleus is not found in green algae, fungi, bacteria, lichen. The answers are: what is powerhouse of cell? Mitochondria. What is suicide bag of cell? Lysosome. What is synthesized in ribosome? Protein. What is physical basis of life? Protoplasm. But here you will have to consider it. If the options are having protoplasm, you will have to tick on protoplasm. But nowadays, not protoplasm, but the DNA is considered the physical basis of life. So if in the options you have a DNA, so you will have to make a tick on the DNA. True nucleus is not found in bacteria. So this way you can make a practice of whatever you have studied. Now let us come on the next cellular organelle, the cell membrane. Cell membrane, which is the outermost boundary wall of the cell. The first and foremost important point of it that in most of the books and most of the teachers teach you that difference in plant cell and animal cell. Both animal cell and plant cell have a cell membrane or plasma membrane. Plant cell, animal cell, both have the cell membrane or plasma membrane. And In plant cells, we have another covering, the cellulose cell wall. Which is not found in animal cells. But in very few books it has been mentioned that in animal cells also, around this cell membrane we have extra covering. Of course it is not made up of cellulose, but it is made up of many other chemicals, and the name is different in different animal cells. Sometimes it is called pellicle, or sometimes it is called basement membrane. So the one very important point is that in animal cells also, around the cell membrane we have extra covering. Of course cellulose cell wall is absent, but in animal cells also extra covering is found around the cell membrane, which is called pellicle or basement membrane. And you would like to know one book which is taught and studied for cell biology all over world. It has been translated in almost all languages: English, French, Spanish, Japanese, Hindi, no Hindi. It is Cell Biology by D. Robertis. It's a very good book. So another important point is that a boundary wall of our lawn or boundary wall of our house is not only a boundary wall. It has certain gates through which we can come inside or go outside. The unwanted persons are not allowed to enter. The inhabitants of that house are kept safe in the house. They are not allowed to go out unless there is a need. So same thing holds good here in cell membrane also. The cell membrane, along with making a boundary wall, it also permits the entry through it both the wanted and unwanted materials. After many discoveries done in 100 years, starting from Overton in 1902. In 1951, the first model was given by Davison and Danieli, which is called the sandwich model. Like a sandwich in which you every day enjoy it, covered from both sides by a bread, and in between there is a packing of butter, jam, or some fruits. Here also the cell membrane is also having a sandwich-like structure. It was given by Davison and Danieli in 1953. So it is the Davison-Danieli model of cell membrane, which says that there is two layers of packing in the middle, the phospholipid bilayer, and the both sides are covered by protein, the outer layer also and the inner layer also. And in the same, as I told you, in plant cells along with plasma membrane there is a cell wall. Here in animal cells also we have extra covering of pellicle. At certain points the cell membrane may have outpushings called microvilli or inpushings called pinocytic channels or pinocytic vesicles. But a few years afterwards, we came to know that this Davison-Danieli model is an oversimplification, and some more properties are there in cell membrane which have to be explained by a fluid mosaic model was given which said that the there are two types of things. One, butter, which is solid, and other is ghee or oil, which is semi-solid or fluid in nature. Later on it was found out that this portion of the lipid present in between is having both types of molecules, some like ghee or butter and some like oil. So they allow the passage of molecules through this. Secondly, it was also known that when this lipid bilayer was frozen, you must have seen that in summer season you take out the butter, it becomes semi-solid, ghee-like. At that time what you do? You just keep it in deep fridge and then it hardens. And then you can cut it in whatever shape you want. So this part of lipid was cell membrane was frozen and then cut or fractured. And then it was observed under electron microscope. It was found that it contained a mosaic floor-like structures having granite like two types of granules. Some granules were smaller and some granules were larger. It was a new thing because earlier we were knowing that the middle portion contains only one type of molecule, the phospholipids. But this study, the fridge fracture study of lipid layer by electron microscope, gave the concept that this layer contains both lipids and some proteins also. So the proteins, along with making the outer and inner layer, they make certain channels also in between through which the molecules can pass through. So there were two things. There is a mosaic-like arrangement in between the phospholipid and protein molecules. And second thing, it had some fluid-like molecules also, so that it allows the passage of molecules through it. And it was given in 1968 by Leininger, which is called the fluid mosaic model. Second point was asymmetry of the phospholipid bilayer. The cell membrane have, along with a boundary wall, it has some doors and windows also, as we just saw. There are two types of molecules. Some molecules are essential molecules. Some molecules are non-essential molecules. So what happens? The environment outside the cell and inside the cell, cytoplasm, is different. At this outer layer, we need that essential molecules should come in. They should be allowed to enter in the cell. The non-essential molecules should not be allowed, right? And just opposite to that, from cytoplasm to outside, the essential molecules are to be detained. They should not be allowed to go out. But the non-essential molecules should be allowed to go out. So, this layer, inner side of the cell membrane and the outer side of cell membrane, both have to perform entirely different functions. And for that, it was suggested that both layers of cell membrane are different, they are asymmetric. Here there are two layers, but the outer layer of phospholipid contains some different molecules, the inner layer of phospholipid contains some different molecules. So both layers are different from each other. Similarly, the outer protein layer is different from inner protein layer. So this concept is called asymmetry of the phospholipid bilayer. Now comes the function. that how the cell membrane allows the molecules to pass through it. And for that, there may be five systems, five methods for passage of molecules through the cell membrane. One is fusion, second is the first system was diffusion. Diffusion is movement of molecules from higher concentration to lower concentration. For example, you just lit an agarbatti in a corner of your room. What happens? Initially, the concentration of the perfume molecules is localized in the corner of the room. But after some time, what happens? All these perfume molecules become distributed all over in the room. Similarly, here also, some of the molecules have a tendency to spread from the higher concentration to lower concentration. So this movement is due to concentration difference or concentration gradient. The second thing is osmosis. In osmosis is also diffusion, but it is a diffusion through a semi-permeable membrane. Semi-permeable membrane allows the passage for some molecules but does not allow the passage for some other molecules. Suppose we have sugar solution. Here we have 3% sugar solution and here we have 1% sugar solution. And this semi-permeable membrane allows only the solvent molecule, the water. The dissolved substance, sugar or salt, is called a solute. So a semi-permeable membrane allows the passage for the solvent molecule only, like a filter. It does not allow the movement for solute or solid particles. Now what will happen here? Here we have 3% sugar solution, here 1%. So concentration of water will be, here it will be 99%. And here it will be 97%. So the concentration of water is 99% here, 97% here. So some of the water molecules will pass through it. It is called osmosis. Of course, the cell membrane is not simply semi-permeable, it is selectively permeable. It allows the movement of some of the molecules, some of the solvent molecules, some of the solute molecules. And it does not allow the movement of some solvent molecules also and some solute molecules also. So this is called osmosis. The third method is active transport. Active transport is energy linked transport. Suppose you are driving by a cycle on a slope. There may be two things. You are not using the paddle and just due to the slope you are coming down. So there is it is diffusion or osmosis. But sometimes when you use your cycle paddle, either coming upward from lowerwards or from lower surface to upper surface, you are using energy. So active transport is energy linked transport. The energy is given by ATP. Mind it, active transport may be of two types. Sometimes energy is used for going upwards. This is normal active transport. But sometimes you are coming down from top to the bottom and you are using the energy, you are using the cycle pedal. It is also an active transport, but because it is an energy linked transport, but it is called facilitated diffusion, facilitated transport. Two more methods are there. One is phagocytosis, and fifth one is pinocytosis. Phagocytosis is cell eating. So suppose there is a cell and some food particle comes in contact of it. So what happens? Here the cell membrane invaginates. And this food particle will come here. After some time, what will happen? Both these layers of cell membrane will bind with each other. It will make a food vacuole. And in that food vacuole, by using lysosomal enzymes, that food is digested. So it is phagocytosis or cell eating. Similarly, pinocytosis is cell drinking, in which suppose there is some water or any liquid droplet near the cell surface. The cell membrane invaginates in the form of a tunnel and that liquid droplets comes in. And then both ends of the membrane unite, it becomes a part of the vacuole. So it is pinocytosis. So this is about the functions of cell membrane, that along with making the outer boundary wall, it also permits, its structure is such that it also permits the entry and exit of molecules through it. If you have any question, you can ask anytime, either by SMS or WhatsApp or Skype software or telephonic call, and I'm always to help you. For some of the students which are studying vertebrate zoology, I have written a good book from Silver Line Publication, Allahabad, which covers the complete syllabus of BSc Part 2. It is a unified zoology which contains both type study, chordates, physiology and biochemistry, even embryology, evolution, etc. So if you need to read a Hindi medium book which contains the technical terms both in Hindi and English, and after every question a summary has also been given, this book can be help to you. Rest tomorrow. Thank you very much.